Jai Sri Ram, Jai Sri Ram. In this video, we will discuss structure of peptidoglycan. So you all know what is peptidoglycan. So peptidoglycan, we all know, guys, that peptidoglycan is something that is forming cell wall of the bacteria. We all know the bacterial cell wall, is very complex, and that bacterial cell wall have different molecule, in which a major molecule, is called peptidoglycan. Either you call it peptidoglycan or you call it murine, right? Now this murine, is making the cell wall of bacteria is quite unique. It will, because of presence of this peptidoglycan and cell wall only, the our bacterial cell wall, make their cell resistant to osmotic stress. Yeah, they protect from the osmotic stress. Fine. Okay, another thing, now this peptidoglycan, it have different thickness in different kind of bacteria. Like if you'll see the gram positive bacteria, in gram positive bacteria, the thickness of peptidoglycan layer in the cell wall, is nearby 20 to 80 nanometer. Okay, now this is about gram positive bacteria. In gram positive bacteria, peptidoglycan layer is thick. Okay, now we will see the gram negative bacteria. In gram negative bacteria also peptidoglycan layer is present, but that will be just 2 to 7 nanometer thick. Gram negative bacteria. Now if we'll see the peptidoglycan murine structure. So murine structure, is how actually it have two derived sugar. One is N-acetyl glucosamine. N-acetyl, NAM means N-acetyl glucosamine, and then NAG, N-acetyl glutamic acid. N-acetyl muramic acid, N-acetyl glucosamine. Fine. So when in N-acetyl, you see that their structure is quite resembling, both are derivative of glucose actually. When in this N-acetyl glucosamine, you see the structure. Actually here acetyl group present at nitrogen actually this is usual glucose, but in glucose, if this is first carbon, this is second carbon, this is third carbon, this is fourth carbon, fifth carbon, and sixth carbon. Here also first carbon, second carbon, third carbon, and then fourth carbon, then fifth carbon, and then this is sixth glucose. Which kind of bond present between this both derived glucose? Beta 1,4 glycosidic bond present. Beta 1 and this is fourth carbon 1,4 glycosidic bond present. Right. Now at second glucose, here one amino group and one amino group present, and then acetyl group present. That's why we'll call it N-acetyl. N means indicating this nitrogen, N-acetyl glucosamine. Okay, when in this N-acetyl glucosamine one lactic acid group will be added, then it will be called N-acetyl muramic acid. So what is difference between N-acetyl glucosamine and N-acetyl muramic acid? N-acetyl muramic acid is a N-acetyl glucosamine with one lactic acid group. Now come back to the structure of the peptidoglycan. In peptidoglycan, there is a criss-cross chain of N-acetyl glucosamine, N-acetyl muramic acid repeated. It's repeating again and again. And then this lactic acid at lactic acid group, actually in the lactic acid carboxylic acid group, one tetrapeptide chain present. Tetrapeptide chain means 4 amino acid long chain present. This 4 amino acid long chain, is quite different than the any other one. They have one L-alanine, then D-glycine, D-glutamic acid, and then L-lysine, then D-lysine. Are you observing both kind of amino acids are present here? L and D, both kind of amino acids are alternatively present here. L-alanine, D-glutamic acid, then L-lysine, and then D-lysine. Right. Sometime at L-lysine at third position, mesodiamine pimelic acid is also possible. So this is one tetrapeptide. present is connected with the carboxyl group of the lactic acid present in the N-acetylmuramic acid, and then this tetrapeptide, it is observed that now this tetrapeptide, you know, side by side they are linking with another tetrapeptide. Yeah, obviously one more I told you that crisscross arrangement, crisscross link is present. So this is one N-acetyl glucosamine, N-acetyl glutamic acid and tetrapeptide, and they are linked by students, and they are linked by side like a crisscross with another tetrapeptide, which are linked by N-acetyl glucosamine and N-acetyl muramic acid. Fine. This direct linkage between two tetrapeptide, yeah, is called peptide, is called, I mean, it's possible in two way actually. What I am saying exactly is possible. This crisscross arrangement, you know, is possible in two way. In one way it is directly connected, and in another way what will happen, same structure will be present, but they are linked by a polyglycine chain, right? One penta chain on 5 chain, one small chain of the glycine in which 5 glycines are present, right? They will be connected like that. Okay, one d-alanine will connected with the L-lysine by polyglycine chain, and this small polyglycine chain is called peptide interbridge. Yeah, we'll call it peptide interbridge. Now the reaction that form this, the form this cross link, see this is cross link between the two, I mean this is one N-acetyl glucosamine, N-acetyl muramic acid, and then their tetrapeptide chain, and then here the link form is actually, it's called cross link. I'm writing here cross link, and if they are connected by polyglycine, then we will call it peptide interbridge. So in this two way they are connected. Okay, two way, this two the tetrapeptides are connected. Fine. Now the reaction which form this peptide cross link, yeah, this peptide cross link during peptidoglycan synthesis is called transpeptidation. Yeah, we will call it transpeptidation. Transpeptidation is very important, very unique phenomena. We will see that in detail in another video. So finally we summarize structure. N-acetyl glucosamine, N-acetyl muramic acid present. The N-acetyl muramic acid have one tetrapeptide chain present, which is linked by another tetrapeptide chain which have their own N-acetyl glucosamine and muramic acid. So they have some kind of crisscross kind of arrangement. Now two possibilities are there. If it is possible that one d-alanine will linked with the second amino acid of the another one by directly, that is called cross link. This formation of cross link is called transpeptidation, and sometime they will be linked by, you know, one small chain of the glycine, which have total 5 glycine amino acid chains are present. In many species, you know, there is possibility this type of, in this peptide interbase, the type of amino acid will be different. That is also possible. Remaining we'll see in next video. Bye.